Thank you. We now have time for discussion. And Jen, I should tell you, I can't see the questions that are coming in. So if you could watch those, that would be good. And I will turn my video on as you suggest. Where is it? Start video. Okay, so my video's on. And as I said, does anybody have any questions? It was a fascinating discussion. I mean, we're all interested in getting older people properly into trials, but if it doesn't work, if they can't attend, if they don't go, then it's not gonna. And these approaches of modeling and using community facilities and all those things are probably ways around that. And it'll be very interesting to see how they work on things like compliance. I just wanted to briefly mention something that came up in the previous discussion. FDA does have a fair amount of guidance, and there's ICH guidance on the same thing, calling for a reasonable distribution of age in trials. People were asking if we've ever sort of insisted or turned drugs down because they didn't do that. And I have to tell you, I have to look into it because I don't know whether we have. But we certainly have many, many instructions to our reviewers that urge them to make sure there are no, what we like to call, stupid exclusions. We don't want people excluded because of concomitant drugs, concomitant illness, or any of those things, because that keeps you from finding out what it is you have to learn about how the drugs work in the population that's going to use them. So we don't like those, and we try to discourage them in interactions with drug companies before the meetings. What I can't tell you is whether we've ever said, no, you don't have enough old people in the trial, and this drug's going to be used in them, therefore go back and do some more data, and we're not going to approve you till we do. I believe that's a possibility, because we would say that your safety database isn't adequate. But whether we've actually done that, I don't know. And I will go looking into it. So with that, any questions people want to raise? This was very interesting about the practicalities of doing these studies. And I was impressed with the difficulties that you had, Steve, in the first study you described, which was famous. I mean, you looked at over 1,000 people and you randomized 18. That's moderately discouraging, and not for something all that obscure or all that difficult. So what did that teach you about all this? Keep things simple?
Yeah, that was one of the points, and also to engage people, and help older people in particular get through the process. So having, I think the earlier trial really suffered a lot by having so many steps that people had to go through. And when you're not capturing people in a clinic, it's a little more difficult to see that they do. But having a relationship, in this case, establishing a person you could call, getting to know their name, and helping them get through each step, calling them if they got stuck, it really helped overcome that. But in the most recent example that I'm talking about, the best thing to do is just eliminate the steps. I mean, establish a relationship and get the essentials done, and then move straight into randomization and treatment. Best way then to help people get through steps is to eliminate them.
So it's really sort of to simplify all aspects of it once you find willingness. But even obtaining consent was difficult, which
actually, so Bob, actually that wasn't the consent process works really, really very well. And in part because we use a lot of audiovisual aid so that people can get much better sense of what they're going to participate in than they do if they're reading a long text document.
Okay, so, I didn't remember your numbers. I thought that for the fraction that consented was fairly small.
Yeah, there were more things involved in that step. including establishing your identity for the purpose of consent. So yes, every time you have a step that means you lose connection with the participant and they have to come back on, that's an opportunity where they lose their attention, lose their connection. Once they get started with the consent process, getting through it is actually pretty successful.
I see. Okay.
With online consent.
That is good to know. But your numbers were scary. We'd heard about them before.
Yeah, no, it was very, very discouraging. But yes, I think the more recent experiences like Topaz and the adaptable trial, the aspirin trial, that we might hear more about from Robert Califf later on. I think examples where simplicity is much more successful in getting things done at home.
Yeah, the aspirin trial sounds a lot like the Harvard Health Plan trial from a million years ago, where it was aspirin versus no aspirin, all done by mail. That was 35 years ago.
Yep.
Jen, you forwarded a question to me, but I can't see it. Maybe you can show it to everybody. Before invitation to a clinical trial, before informed consent, an educated patient would be, and I couldn't read the rest of it. Can anybody show it? Maybe I could find these somehow.
Can you hear me okay?
Yeah, that's good. Why don't you just say it?
Yep, I can go ahead and share it for you. So the question is, and this is from one of the planning committee members, is how well does the predictive modeling account for multimorbidity, such as synergistic effects, beyond the summative effects of the individual conditions?
Okay, Christina?
Yeah, I can definitely speak to that. Can you hear me okay?
Yes.
Yes. I mean, it's a great point, and it's one of the key reasons why we do mechanistic modelings, because biology is not linear. You absolutely have to account for not just the aggregated effects on a particular on-target effect, but also downstream feedback loops. So yes, it can get very complex, but that's precisely why we would want to have a mechanistic model that can account for those interactions in a more thoughtful way. I can't really say more without knowing exactly the question that you have in mind. But in general, I would say the mechanistic models are very well suited to looking at these more complex relationships that are really quite difficult to tease out of just empirical modeling or statistical modeling on clinical data. You never have enough granularity in just the clinical data to really tease that apart. So we bring in what we already know about the mechanisms to do a better job of extrapolating beyond what's already been established.
So these models are used to help you design the trials, right? That's
what they can be. Yes.
Okay, I understood how you can use PK information to do that. That's sort of clear enough. I didn't understand where you got your pharmacodynamic models to include in those things, and those would affect patient population size and all that stuff.
Yes. So the PD effects are, we start with the representation of the actual disease pathophysiology. So we'll include pathways that are known or hypothesized to be involved in the disease, not just pathways that have previously been targeted by different drugs, but we really look at the entire literature to get a complete picture of the overall orchestration of the disease state. And then that's regardless of any drug effects. Then the drug effects are introduced on top of that to say, okay, which pathways do we know the different drugs are interacting with? And again, there we can draw on literature from existing drugs. If we know a particular existing drug's mechanism of action, and we have some clinical data as to how well that drug worked, what its effects were on different biomarkers, et cetera, that's extremely helpful for allowing us to refine the overall model of the pathophysiology. And then the new drug that we're interested in, is introduced into the biology, again, by its mechanistic effect on those pathways. And then the model will basically, simulate out the ripple effects from interfering with that particular pathway all the way down to the clinical endpoint that we're trying to predict. And when I say the model, there's many versions of the same model, which we call virtual patients. So you can look at how differently do different patients respond to that same drug. So the PD model really comes from the entirety of the existing literature. So it's not so much just looking narrowly at the particular drug that you're investigating, but really first representing the overall mechanism and then introducing the perturbations that the drug is expected to affect on the disease.
So these models are used to try to figure out what doses to study in the elderly and how often to give them and stuff. Have you actually looked to see where people made use of those models and then see whether it really worked out? And a question from somebody was, we make adjustments for dosing in children all the time. Not that I think we necessarily know how to do it, but I think we usually do it because of their size, not because we think the drug works differently in children. It seems very hard to know from these data whether the dose would be different in adults. In most respects, you might, if you knew the drug makes you dizzy, you might say, I'll use a lower dose in adults. We've had that example before with sedative hypnotics and stuff. But where else can you actually use those data to adjust the dose? Other than PK, I get that.
Oh, sorry, was that, I'm not sure I follow can you repeat the question? Was that addressed to me?
It's addressed to you.
Okay, I'm sorry.
But it also reflects a question that somebody wrote in.
Okay.
That we adjust dose for children all the time. But frankly, I think that's mostly by size or by
yes, it is. That's right.
And things like that. Not usually for pharmacodynamic differences.
No.
And I'm interested in where we've been able to actually do that for a pharmacodynamic difference.
Yeah, I'll let others speak to where that's actually been done. I would just advocate that. I think it should be done a lot more because children are not just small adults. There absolutely are some very important differences. And I think the modeling can help, get you in the ballpark of where you should be dosing in children. If again, the example I gave, there were some significant differences in the reproduction rate of the tumor or the malignant cells, as well as the maturity of the immune system. That would absolutely be expected to have an impact on the.
It would really be interesting after making some of those assessments to go and look at the concentration response relationship in the older population and compare it to the younger population and see whether it predicted it right.
Sure.
Jen, there was another question and I've forgotten it already.
Yep, no worries. I've got it right here. So
okay.
Another question was, could Dr. Chen and Dr. Berry please discuss how pharmacy might be part of adaptive designs to reduce dropout and perform adaptive randomization or record adverse events?
Scott, do you want to start with that one or do you want me to go first?
Sure, I'll touch on the back part of this. The logistics of having the pharmacy do it, I think you could absolutely do that. There's the operational aspect of getting the dose to be done. At the very beginning, you could construct an adaptive design where you have different doses being given to, I'll dichotomize it by saying not old and old populations, but you could even differentiate it into further classifications to be assigning different doses as a function of efficacy and safety for those populations, and the design would create those randomization probabilities. Now, operationally, sure, I imagine, and you can describe how that might be done, Steve, at the pharmacist level.
Yeah, I'll try to speak more to the practical nuts and bolts and how this works. A standard of care that pharmacists provide in the space of medication management is comprehensive medication management. And the way it works for seniors is you naturally start with a much lower dose, but the important difference is pharmacists are seeing these patients very often. They're checking in sometimes after a few days, after a week, at frequencies that are very difficult for really almost anybody else in the healthcare system to do. And by doing that, we catch very early on adverse drug reactions or potential problems with the medications and avoid having patients go down that path, actually suffering from an adverse drug event. So I think that's a very valuable reason why engaging pharmacists in this work is so helpful. It's because of that extra layer of monitoring and frequency of monitoring that occurs when they're involved. Some of the other questions that had popped up within that space are, do pharmacists actually record these things? They have to. They need to, and they are required to log ADRs in whether you're in a hospital setting or in a pharmacy setting. And as I mentioned before, there are platforms now commercially available that do more than that, right? Pharmacists can now track all the drug-related problems they identify and resolve and aggregate it in a standard national format. There's something called SNOMED CT codes that standardize how all that's reported. How would this help to reduce dropouts? Again, because of that extra layer of monitoring, extra layer of safety, we can help ensure that that participants don't suffer serious adverse events and that they're caught early and managed early. And by the way, I'll just add that most of our work is done doing propensity score matching. So we don't so much randomize as we identify matched populations and compare using that approach.
Just a quick comment on something you said, Steve. This is Scott again. There are two types of sort of adaptive randomization. One is based on previous patients. It looks like one dose is better, so we'll assign them to the dose. Another one is a strategy that alters a dose within a subject. So if they're doing well or not, you could increase or decrease the dose. That's a different strategy within patient to alter their dosage.
Jen, you sent me another question, but I can't read it.
No worries. That's what I'm here for. So Another question is, someone is asking about a particular challenge that wants the challenge to be addressed right now by this panel. If someone could address the challenge that while there are not good pharmacodynamics data on changes in all systems in older adults, while models still require assumptions. I don't know if that's clear. I can try to rephrase it here possibly. I think the challenge is that there are not good pharmacodynamics data on these changes for older adults, but models require these kinds of assumptions. So I think the question is how to deal with that challenge.
Yeah, that's not unlike the question I was trying to ask before. Where do we get these models and how credible are they? And in the end, do they work out? But, right.
Yeah, I can make a comment on that. So I think it's important to set the appropriate expectations for what the model can do. And certainly, if you're in a data poor area, I would not try to rely on the model to give you the precise prediction of exactly what will happen in each type of patient. But you will always be, have a better understanding of the issues with the model than without the model. And the model can at least help you anticipate those aspects of the variability due to some of the factors that we talked about that can be anticipated. So the model allows you to put together what you currently know. It also allows you to really identify what we don't currently know. We do a lot of sensitivity analysis, and we like to predict not one point estimate, but a range of possibilities, which is still better than, just operating completely in the dark. So I think as long as you're careful about being very upfront about what the model can and cannot do, you will still learn something. And in fact, a very useful way to apply these models is to identify what aspects of the physiology would be most important to learn more about to reduce the uncertainty. So we do that all the time, where we basically use the model then to identify what additional experiments or biomarkers to collect at the next clinical trial would be most useful for better understanding what's going on.
Can you use data from the developing trial to modify your model? Or do you?
Yes, absolutely, sure.
Yeah.
Mm-hmm. I think it's really, ideally the modeling and empirical data collection are iterative. So the model's always evolving. So again, we're not necessarily trying to predict all the way to the final endpoint, but we're trying to help you take more informed steps along every step of the way.
Are there any specific wonderful examples of where this has in fact worked, where there were major modifications to an older person trial that were based on these things?
Okay, so I personally am not aware of these models having been used for older populations. We do have some pediatric examples that I could point you to on our website, rosaandco.com. But yes, I think that the same principle applies, but absolutely.
And where the models, we all understand how knowing pharmacokinetics can lead you to change things, but are there some good places where the PD aspects of it really led to changes in the dosing strategy and things like that?
Yes, absolutely. So for example, just being able to, a recent example was shortening a trial from 12 weeks to 4 weeks, because we could demonstrate pretty conclusively that all the timescales involved for the mechanism of action and the effect on the efficacy, that all plays out in the first couple of weeks. So you don't have to run a 12-week trial, you can run a 4-week trial. That's a simple example, but certainly we have many other examples.
I see a few more questions. Heather, want to say what it was? Or Jen or somebody?
I can share that with you. So the question was, could we learn a little bit more about how the Remap design works for embedding in clinical settings, for example?
Sure. So Remap is an acronym for randomized, embedded, multifactorial adaptive platform. So it's several of these are being used. One is being used in community-acquired pneumonia, where multiple therapies are given to the individual, randomized multifactorial across therapies. The goal is to be embedded within the healthcare system. You're randomizing them within the healthcare system. You're providing them the order sheets for this. You're pulling the outcome data from the system. So this seems like an ideal thing to do within a healthcare system. I think the pre-approval of things in the FDA is one situation, but suppose there are multiple drugs out there and we need to learn post-approval comparative effectiveness, treatment heterogeneity. You could embed multiple therapies within a healthcare system, randomize those therapies, continually update which ones are providing better treatment outcomes, safety, and efficacy. That's really the goal of these. The Remap CAP trial was brought to community-acquired pneumonia that then expanded into COVID-19. So it's investigating 6 different domains of therapy, adaptively randomizing across the therapies, and much of this has been embedded within the healthcare system, which is needed within a pandemic.
Okay, Jen, I see another question.
Yep, I believe we'll probably have time for maybe one more question. But we've got a couple more here. It's hard to cut it off. So the next question I received was, since these models have so much complexity and so many moving parts, and there are relatively few measures taken in clinical trials, how do we know which aspects of the model to update? It seems that, it seems to be that it would be highly underdetermined unless we measure everything the model predicts.
I think that's another question for the mechanistic modeling. So, agreed. Yes, you can never measure everything. So it's a longer conversation, but any clinical biomarkers that we can get are always very valuable, but they are represented in these models in in the context of, for example, the different cell types that are producing that soluble marker, or basically any other aspects of how that biomarker comes to be expressed is included in the platform, so in the model. So if you see a particular effect on a biomarker, we're not trying to interpret that signal in and of itself as being indicative of something. We take that signal back and say, okay, so what could this mean? Did our model predict a similar pattern of biomarker response? Why did it predict that response? The beauty of the mechanistic model is you can always dig in deeper and look at why is the model producing that similar effect. If there are differences between what the model would have predicted and what is seen in the clinical trial, again, we would unwind that and try to understand why. So this is really, it's not one piece of data. It's a piece of data in the context of this whole model of the physiology that we've already created. And it's a biological investigation at that point to try to understand, what is actually leading to that expression profile or whatever the readout is in the patient population. So you're right, the models are underdetermined if you purely think of, each individual piece of data, but the models are actually quite constrained when you think about the overall biological picture and what we know about biological mechanisms as a whole. So it's a much longer conversation that I'm happy to have offline as well. But that's the basic idea, is that you don't consider the data just by themselves. You consider the context, the biological context for all of these signals.
Okay, does Janice get to ask her one last question?
Yeah, we can go ahead. We're going to have to be pretty quick here, so we can wrap it up and pass it off to James, but maybe we can squeeze one more in.
She's asking about off-target toxicity.
Yep, so I'm seeing an IA challenge in older adults is how to predict off-target toxicity in models. We are doing trials of new drugs that we cannot really be certain of effects in patients, especially a heterogeneous population. Not sure there's a question in there. Let me just check here above in the chat here.
Well, for those, that's why you have to actually do the study eventually, find out about those things.
Yeah, so, we've got other questions, but I think we're actually a little bit low on time, so we might want to pass it off back to James here. But just want to thank you all, and I'll hand it off to you, Bob, to close and pass off to James when you're ready.
Yeah, I'm glad to. This was a very satisfying discussion. The practical the practical necessities and opportunities in this area are very numerous. I mean, even if you do manage to enroll older people, you still have to figure out what the differences are likely to be and design the studies properly. This was a very nice discussion of all those opportunities. So thanks, everybody.